Hello, my name's Katie Pickup and I'm a reviews editor at the Company of Biologists. We're the publishers for Development, Journal of Cell Science and Disease Models and Mechanisms, based in Cambridge in the UK. It's really nice to be here at Cold Spring Harbour for the 89th Symposium this year on senescence and ageing, and I'm very excited to get to talk to Jesus Gil from the Laboratory of Medical Sciences in London. So could you please start by introducing a bit about why senescence is a problem in ageing and cancer?
Yeah, so senescence itself is a stress response that is protective, but what happens is that during aging, in cancer and in many diseases, senescent cells accumulate aberrantly, and that aberrant accumulation, what it does is cause inflammation and cause disruption in the tissue, and it has been shown that it contributes to aging progression and to cancer and to very different diseases related with aging.
Yeah. So you're looking at trying to eliminate these senescent cells to try and improve disease outcomes and cancer. So could you explain a bit about the strategies that you're using to do this?
Yeah, there is quite good proof of principle showing that if senescent cells are eliminated, you achieve benefits, at least in preclinical models of either aging or cancer or disease. And we are trying to eliminate them in different ways. So on one hand is trying to use senolytics, that are small molecules that selectively kill senescent cells. And we have been doing different screenings to try to look for some of these molecules. On the other hand, we have been also trying to see whether, rather than killing the cells, we can just modify the phenotypes that are like detrimental. For example, we have looked for ways in which we can just suppress what they are secreting, suppress this ASP, or even decrease the entry into senescence. And finally, the thing that we have been working lately, and also many other groups have been working, is try to exploit the natural mechanism that eliminates senescent cells, that is the immune system. So we are trying to see whether we can just potentiate the immune-mediated elimination of senescent cells.
Yeah, that's really interesting. So the senescent cells have this senescence-associated secretory phenotype, the SASP, and that you're trying to enhance that, right, to try and get them to be targeted by the immune system. It seems sort of paradoxical in some ways that you're trying to make them more senescent in order to get rid of them. What are the risks of doing this as a treatment?
This is one of the strategies that we are just trying to do. Try to see whether we can invoke the immune system to try to eliminate the senescent cells more efficiently. So that's what happened naturally. Yeah. And naturally the senescent cells are secreting this SASP, so this bioactive secretome that can attract the immune system to eliminate them. One of the things that we are trying to do is to see whether we can potentiate this ASP and then try to attract more efficiently these immune cells and then contribute to the elimination. I think this can work in the context of cancer, for example, but of course, before I just explained that this ASP is also contributing to some of the detrimental effects of senescent cells. So we have to do that in a way that is controlled. And as all of these strategies, we have to see what are the benefits, that we have seen that there are some benefits, but also try to be wary of what can be the side effects of employing these strategies.
Yeah, and it seems quite nice that this is quite specific to the senescent cells and you can target them quite specifically without harming other cells in the tissue or the cancer. So because you showed some really nice data with using it in combination with cisplatin chemotherapy. Could you explain a bit about why this has promise in cancer treatment?
Yeah, so one of the things that we try to do is to try to use this strategy to enhance the elimination of senescent cells in the context of cancer. So we went to ovarian cancer, that is a cancer that's of unmet need. So now the standard care is cisplatin. Cisplatin, we know that induces senescence. And then what we try to do is to see whether once that we induce this senescence, we can potentiate the elimination of the cancer cells by potentiating the recruitment of the immune system. So we came with a molecule, a PROTAC that degrades SMARCA4. And when we do that, so they synergize into the elimination of the cancer senescent cells. And one thing that is interesting is that in ovarian cancer, so immune infiltration is predictive of good responses, but paradoxically it has failed. The clinical trials on immune checkpoint ligands inhibitors, they have failed. So there is the knowledge that if we potentiate the immune system, that might be beneficial, but there is the need of trying to do it in different ways.
Yeah. Do you think, or does the field have much of an idea why there's a lot of these drugs and things that are showing so much promise in vitro in animals, but not so much in humans?
I mean, being fair, the senolytic field is quite nascent. I mean, the concept of senolysis itself started probably 2011, 2016. There was like the paper that saw it on the context of natural aging. First clinical trials 2019, and we have just a bunch of these clinical trials. If you compare that with cancer, with immunotherapy, that there are like hundreds, if not thousands of trials, I think they're still in the infancy. Said that, so there is like very nice proof of concept data in preclinical models, but translate that is not always easy, and particularly we have to try how to kill different types of senescent cells, also understanding what are the regimes and the timing for killing these senescent cells, not overdoing it, do it at the right time. So I think it's complicated, but I'm hopeful that despite that there is no success yet in the clinical trials, that the concept is there, and if we do it right, so we can have some success.
Yeah, definitely. It sounds like a bit of a challenge, but yeah. So you presented quite a lot of nice understanding of the mechanisms of why getting rid of SMARCA4 actually helps this. What do you think? Because a few people have been talking about sometimes, okay, if a drug works and if you see a promising phenotype, does the mechanism matter? So what are your thoughts on trying to find the mechanism?
Yeah. I mean, in general, I come from the basic biology angle, right? So I started studying senescence more than 20 years ago, and I was interested in understanding how the cell cycle arrest is controlled and how the SASP is controlled. And what has happened with time is that we have realized that all this knowledge that we have might be now useful to try to translate it into potential some therapies. I think that at the end, being pragmatic, if a medicine works, and I think that, people are not going to worry what's the mechanism, but understanding the mechanism might help to, for example, stratify the treatment in clinical trials, understand also whether the patients are responding or not, and all of those things are essential for the clinical trials to be successful. And at the moment, I think that we are in the state that we have to try to make them successful to prove at least that the concept is worthy, and that will spearhead like more of these senolytic treatments. Yeah,
I think it seems kind of safer to at least know what you're working with.
Yeah.
So what do you think are maybe the biggest challenges facing the field at the moment? Well, I guess the translatability is one, but
Yeah, I mean, a challenge in the senescence field from a long time is understanding what is senescence, right? So it's probably senescence are many different things for many different people, and also it's a very heterogeneous state. So different cell types undergo senescence in different ways, and then to try to define, and not just define, but basically characterize, detect these senescent cells is difficult. So detection of senescence, understanding how these senescent cells in vivo behave, I think that is a key challenge. This is essential not just for the basic knowledge, but it's also essential later on to try to make sense of the clinical trials. And of course, I think that the other thing is try to see whether any of these promise that we have in preclinical models can be translated into different models of disease.
Yeah, I think it's tricky, particularly when it's something that was maybe first observed in vitro and trying to find the elusive senescent cells in vivo. So what do you think what do you hope to see as advances in the field in the next kind of, 5 to 10 years? What are you most excited about?
I think there is a lot of promise on the detection of senescent cells and definition of different types of senescence, understanding this heterogeneity, especially in vivo. And I think that from all that knowledge is going to also come like mechanistic insights into how we can target these senescent cells. So I think that we need like all this field of basic knowledge on understanding senescence, but eventually I think that given the promise that we have right now, I think that it will be amazing if some of the clinical trials that are started go to a point that we can have like these senolytic medicines basically in the clinic.
Yeah. And do you think at the moment the most promising things are kind of using this sort of 1-2 punch approach of treating, like in cancer particularly, I guess, starting off by kind of inducing more senescence?
I think the good thing and the bad thing with senescence is that it almost involved in any type of disease that you look, and that's really good because it has like a broad appeal, but also it complicates where to concentrate efforts. I think cancer is an interesting one in the sense that a lot of the current cancer therapies are inducing senescence, so you can have this 1-2 punch approach where you first induce senescence in the tumor, being by radiotherapy, by chemotherapy, or by some of the target therapies, and then you come with a senolytic in combination or sequentially. This is promising because obviously there is plenty of clinical trials on cancer, plenty of combinatorial trials, and I think that is an easy entry point. On the other hand, if you want to eliminate cancer cells, you have to have like a big effectiveness. If not, the tumor are going to reappear, right?
Yeah.
And if we just flip the argument, what we'll say is in many of the diseases where senescent cells accumulate, being in pulmonary fibrosis, being in osteoarthritis, in ophthalmological disease, senescent cells are a contributing factor through what they are secreting, and maybe we just kill just a small percentage of the senescent cells. That's sufficient to go under a threshold and have an effect on the disease. And then the barrier to have a partial effect that is not going to be effective in cancer might help to try to see something that is effective for the disease. So yeah, I think we have to try to put our eggs in different baskets.
Yeah, I think that sounds very promising for the future of different diseases. Do you think are the clinical trials harder to run in different, because I feel like in the diseases where you've got different tissues and there's less kind of known about all the different interplay with senescence and things? Yeah,
so I think it's coming back to what we were arguing before. I think that having the knowledge of mechanisms, the knowledge also of what cells are senescent in a specific disease or a specific tissue is essential to try to be able to set up some of these trials, understand what are the drugs that work best, and also be able to both stratify the patients that are selected for the trials and also monitor whether they are working or not. I think we need that kind of knowledge, and I think that's of all the possibilities of treating many different age-related diseases or cancer. I think that there are already a number of different clinical trials ongoing, and the expectation, if we are optimists, is that at least some of those will work, and I think that then that will just establish the basis to understand what we can do to improve the trials or try to make some of the other trials in other diseases successful.
Yeah, definitely. I think understanding what's going on to make sure in different contexts and seeing the parallels is very important. Yeah, that's really interesting research. Thank you very much. It's been really nice to talk to you.
Thank you so much.
Thank you.